Carl Flannery, who's the Senior Director of Scientific Affairs at Bioventus LLC. Carl is a molecular biologist and biochemist who has had an extensive industrial career developing medical products for the treatment of orthopedic pathologies like arthritis. He has spent time at Wyeth, Pfizer, and Sanofi before his time at Bioventus. He is an author on over 60 peer-reviewed publications and an inventor on many patents, including seminal discoveries in terms of mechanisms of arthritis. So I thought it would be great for Carl to talk today about their work at Bioventus and developing biologically therapies that are biologically originated for targeting tissue regeneration. Carl.
Okay. I'm going to start my video. There we go, so you can see me. And start the slide. There we go. Larry, can you see my pointer? I'll use that as a.
Yes, perfect.
Okay.
Yes, that's great.
Thank you, Larry. Thanks for the invitation. As always, a great pleasure. Unfortunate, of course, not to be at Cornell to visit you guys there again. And it's always a great experience to meet lots of different people, lots of different expertise. And what I'm going to do today, just get myself coordinated here, is talk about soft tissue biologics. So maybe diverting a little bit from the sort of synthetic aspects that you'll hear in many of the talks today. And we'll actually talk about a tissue, a natural tissue that we have been developing for use as a therapeutic in osteoarthritis in the first case, but should have utility in a number of different pathologies. We're mainly focused on orthopedic pathology. So we call this MOTYS, also PTP001, and we'll get used to that terminology as I go along here. Just quickly, folks who don't know about Bioventus, we're headquartered here in Durham, North Carolina, in the Research Triangle Park area. We have our main offices that a lot of our different divisions are located in. We have some offices in Memphis, Tennessee as well. Some of the manufacturing happens there. Sales and marketing in Canada, Netherlands, and other support groups there. And then most recently in Valencia, California, with the acquisition of Bioventus. Our kind of tagline used to be, innovative orthobiologics. And we've sort of branched out a little bit more and recognized that a lot of what we do is treating pain, not necessarily focused on orthobiologics. We have a lot of devices. We also have, for the last couple of years, we've had an active R&D lab in the downtown Durham area. So that's a part of a biolabs facility. So a lot of the work that I'm going to talk about today either was performed or originated in the lab here, the lab team here. So our products, as I mentioned, we have some devices. We have a low intensity pulsed ultrasound device for fracture healing that's called Exogen. We have some surgical augmentation products, which are really for bone healing. We have an allograft product there and some synthetics. And then probably more relevant to what is being talked about today in this symposium, one of our other main product lines are hyaluronic acids. So I've just put one slide here indicating those products. So on the left, we have Durolane, which has been most recently introduced in the United States. That's a single injection cross-linked bacterial origin HA with a pretty extended half-life in the joint. We also have these other more legacy products, Supartz FX, which is uncross-linked, and also Gelson 3. So these are multiple injection regimes, whereas Durolane is a single injection. And many of you are going to be familiar with HA. This is obviously a sort of almost a quintessential soft tissue. Of note, these products are all marketed as devices. That was the original designation when these intra-articular treatments came on the market. We know it's an unsulfated GAG, repeating disaccharide, a very, very simple structure, but has many different and complex functions. It's viscoelastic. You can see in the picture on the bottom there, just pour some of this out or try and syringe this material. It's very, very viscous. Acts as a hydrodynamic lubricant, but also seems to have some boundary lubricant effects as well. It's a major component in cartilage. It anchors proteoglycans, the large aggregating proteoglycan, which is really responsible for a lot of compressive resistance of the tissue. But more and more evidence coming out, and it's been coming out for quite a while, that HA probably has a cellular effect, binds to receptors. CD44 is one of the most studied. There are some others out there as well. So moving forward in the regulatory space, it seems likely that these new products in this category are probably going to be regulated more as drugs than as devices. Anyway, that's the sort of portfolio we have now for treating OA. These are all intra-articular injections, of course, local injections into the osteoarthritic joint. But what I'm going to focus on today is a product that we've called MOTYS, also called PTP-001. It's a placental tissue particulate. So we're sort of going back to the roots of soft tissues, I think. I mean, this is the origin here, this little picture I have indicating a developing fetus. We're all pretty familiar with an umbilical cord, and here as we've been developing this product, we've gained kind of a secondary knowledge of some aspects of OBGYN as well as cartilage biology. So the umbilical cord is obviously providing a lot of nutrition to the developing fetus. And then the sac that surrounds the fetus is comprised of really two fairly distinct tissue types: the amnion, or the amniotic membrane, which is the fetal side of that sac. And then on the maternal side, you have the chorion. So these are fairly tightly integrated together, although they can be peeled apart quite easily when you recover the birth tissue. So we can obtain placentas at the time of C-section birth from consenting mothers. These are healthy mothers. And there have been a number of these types of products that have been marketed as what are termed HCTPs, or human cell and tissue products. And they're really, you know, they have not been regulated as drugs or devices. They're a very specific category. Donor screening is required. They have to be processed in a certified tissue bank. And they really are, these are allografts. So much like the demineralized bone products that are out there, these are donor tissues that are used in an unrelated recipient. So in a nutshell, MOTYS, as I mentioned, it's a micronized tissue particulate. It's terminally sterilized. We've optimized the processing to preserve bioactivity. We're going to look at some of those features in this slide deck. As the combined amnion, chorion, and umbilical cord tissues, it's obviously non-cellular since it's been lyophilized and undergoes quite a bit of processing. So there are no viable cells. And then that tissue particulate can be resuspended in saline for injection. So where we are right now with this product composition that we've been characterizing, and the term that we've given it is PTP-001, is currently being developed as a biologic under an IND application with the FDA for the treatment of pain and impaired function in OA patients. So patients with osteoarthritis of the knee. So if you look in clinicaltrials.gov, you'll find details of our phase 1 trial that's ongoing right now. And really the FDA, there's kind of a sea change going on in some of these allograft products that have been marketed to date, with the FDA really getting much more stringent on definitions of both minimal manipulation and homologous use, which is the definition basically of the tissue product. They must not be more than minimally manipulated. and they must be for homologous use. And so this kind of category of micronized tissues, particularly placental tissues, has come under that scrutiny. And really the guidance from the FDA is that to continue in the marketplace, these will need to be regulated as a biologic. And you're essentially going through the drug development pathway to do this. So as I mentioned, we're developing this initially. The first indication is for osteoarthritis. And this really, again, just to back up a little bit, these tissue products really don't have a label indication. They're much more generalized for use in treating degraded tissues, whereas once you go through that IND and biologics license pathway, you will get a label indication for a specific disease or indication. In the first case, we're going for osteoarthritis. So it's sort of a generic picture here that we can see that, comparing a healthy knee to an osteoarthritic knee, I think most of us possibly have, or certainly know relatives, friends, colleagues who suffer some sort of joint issues. Everything from I've had an injury, I'm probably going to develop early OA, I already have OA, my joint hurts when I'm getting out of bed or going upstairs. So it's a real sort of epidemic in the healthcare industry with no treatment. So it's a slowly progressive degenerative joint disease. Multiple tissues are involved and multiple pathways are involved. And I think, I've been in the field for quite a long time now, and I honestly don't know that we're that much closer to figuring out exactly what it is, other than there are many pathways that are involved. It's not just a single target that's going to be a magic bullet for treating osteoarthritis. Some of the features are that the synovial fluid has, in healthy joints, it has a high concentration of HA and other lubricants, whereas those can change during osteoarthritis. There are obviously tissue changes. Cartilage gets eroded. There's inflammation of the synovium. There are subchondral bone cysts or bone marrow lesions that develop. I think we still don't know where the pain comes from. That's the primary reason that patients come in and see their doctor and then ultimately an orthopedist. Their joint hurts. They have limited range of motion. We still don't really understand what's going on there, at least in my opinion. So back to these placental tissue allografts, of which we're talking about MOTYS. These are pretty fascinating. As I mentioned, they come from healthy C-section births. They have an intact extracellular matrix. There's collagens, laminins. They're a pretty rich source of proteoglycans. They have a lot of growth factors. You can go into the literature and find plenty of studies that talk about that. And so seemed to be, when we first started looking in this area, seemed to be a pretty good candidate as a biological treatment to address a number of the issues that are prevalent in osteoarthritis. So they potentially could prevent inflammation, they could reduce tissue damage and promote tissue growth and repair. One very interesting feature is that these tissues are, just by their very nature, they're immunologically privileged. So they have low levels of cell surface antigens, and as you can imagine, these tissues have to coexist with a developing fetus and the mother as well. So much less chance of a sort of immune reaction in that case. They've been used for a long time in medicine. We heard some talks, certainly this morning, talking about maybe the use of membranous structures to cover wounds, for example. These have been used for a long time in the clinic as wound coverings and to aid wound healing with some very, very good and promising results. They've been used in ocular indications. And they're really more recently, I'd say just within the last few years, have gained prominence in orthopedics and sports medicine conditions. So osteoarthritis has been the use of these particulate preparations from several different companies, used as injectables, but they've been used for tendonitis, plantar fasciitis, even sometimes as a kind of a membrane wrap in the tendon indication. So what we're going to focus on today over the next few slides are just some of the preclinical areas of work that we've been working on: in vitro growth factor and protein assays, some of the bioactivity assays, the cell-based assays we've conducted, and then talk a little bit about the in vivo studies that we've done. And all these studies support the purple boxes at the bottom here are indicating the sort of areas that you need to address when you're working with the FDA to get approval. You need purity and identity assays. And these are fairly common terms in the drug world. And I think what we're doing here, it's kind of breaking new ground, is taking a tissue product and trying to convert that using the same amount of stringency into a drug product. So you've got to have something that's reproducible every time you make a new lot of material. You need good identity assays. You need potency assays that are reproducible and fairly high throughput. And you obviously have to do all the safety and efficacy studies that go along with that approval process. So I just wanted to mention, although we wanted to do this a year ago, it's kind of been a little fortuitous because it's given us some time to develop more data. A lot of the data I'll talk about today has now just been published. It's come out in Osteoarthritis and Cartilage. We've got folks from Bioventus, Scott Seaman, Kelly Budden, they're actually with me today, providing me some local support. Alessandro Pavesios is our Chief Science Officer at Bioventus. We have folks, Mike Nassert, Eric Semler, and Mark Long. They're from MTF Biologics, who's our partner in the development of this product. They're a large tissue bank in New Jersey. And then Jacob Favre at Boulder Biopath doing the in vivo work, and Catherine Kelly and Dr. Richard Loser at UNC Chapel Hill, who we've been collaborating and doing research work with. So in MOTYS, if I kind of put it into a nutshell, those three kind of areas that I talked about: proanabolic factors present in the tissue. We have factors like basic FGF. These are all things that have been talked about, written about, suggested to be very important and potential therapeutic targets or modalities in the treatment of osteoarthritis, and some pretty good data behind them, but all as kind of individual factors, not as a combination type therapy, which I think you can envisage could be part of the potential for MOTYS and these placental tissue products. Anti-inflammatory factors, interleukin-1 receptor antagonists, that's obviously very key in blocking inflammation. Certainly therapeutics have been developed around that, sort of inhibitors of IL-1. And then anticatabolic factors like the tissue inhibitors of metalloproteinases. These directly inhibit some of the catabolic enzymes that we know are responsible for cartilage degeneration in OA. So we took a look at those. We took some of the MOTYS tissue particulates, we extracted it. We did some kind of high level protein arrays to look for different classes of molecules that may be interested in for us to look at. And then we kind of took the next step and used some quantitative ELISAs to do some robust quantification and characterization. These have all been validated by a third party. And what's shown in this graph are 12 different donor lots of MOTYS. So each placenta is a lot that we can derive different numbers of treatments from. On the y-axis is the picograms of analyte per milligram of tissue. And you can see all the factors that we talked about in the previous slide. I think the real takeaway from this is that the individual data points indicated by open circles show us that there's a pretty high level of consistency between placentas. And that's not something you'll typically see in other tissue allografts. So we feel pretty confident that every time we make a different batch of MOTYS or PTP001, we're within a pretty tight range of these different factors, these biofactors that are available there. We then moved on to develop some bioassays here that we could use for screening or characterization of the different lots. One is an anti-inflammatory bioassay using a monocyte cell line. We can expand that. We can differentiate them to become a macrophage-like phenotype, stimulate them with lipopolysaccharide, which causes them to express large amounts of pro-inflammatory cytokines, and then culture them in the presence or absence of the MOTYS eluates. So what we call eluates are just the proteins that elute out of the tissue. And then we can measure those concentrations of cytokines using standard ELISAs again. And some of the data looks like this. So we're measuring TNF alpha and interleukin-1 beta levels. On these graphs here, the gray bar is LPS treatment only. So we're upregulating the production of these cytokines. And then as you add increasing amounts of MOTYS or PTP to the culture system, you can see a dose-dependent decrease in the production of inflammatory cytokines. And then the last bar is always a dexamethasone or steroid control, which has a very pronounced effect in this type of an assay. So very significant results here, blocking production of inflammatory cytokines. We also looked at an anticatabolic assay. So this is work we did with Dr. Loser in his lab, isolating primary chondrocytes from human osteoarthritic cartilage. After joint replacement, we grow those up, stimulate them with a catabolic agent to induce MMP expression. In this case, we focused on MMP13, which is the major collagenase that is involved in cartilage degeneration during OA. Very similar system. We add these eluates from MOTYS, and then we've measured MMP13 protein levels by Western blotting. So that's shown on this slide. Again, just to kind of walk you through what I've simplified it here with the dashed boxes and on these Western blots is that when you add the catabolic stimulus, that's a fibronectin fragment, a catabolic fibronectin fragment, you can dramatically upregulate production of MMP13. And then as you add increasing amounts of the MOTYS eluate, you can see the density of that band is significantly decreasing. And then we've quantified that on the right in the bar graphs. Just kind of focus on the set of data on the right here. When you've added that catabolic stimulus, that's the level you have, so it's normalized to that, the level of MMP13 protein. And then as you add the MOTYS eluates, you can see a very, very dramatic decrease in MMP13 protein levels in these culture systems. And then finally, more of a kind of a workhorse that we do, proanabolic assay. We've just culture expanded a human synoviocytes cell line, again, adding MOTYS eluates. And then we can measure cell proliferation using a pretty simple metabolic assay. And again, similar sort of looking data here. This is viable cell number. Basal media, really not much going on. In fact, the cell number is decreasing. You add MOTYS to this culture system, you can stimulate cell growth and proliferation, indicating potential for anabolic effects. And then this is just a control with 10% fetal bovine serum that most people use in their culture systems. So I think that that's some very encouraging in vitro data. And then what we did is kind of take that to an in vivo setting and see what would that look like in an animal model of osteoarthritis if we treat those joints with MOTYS. So we've used a rat osteoarthritis model here, destabilization of the medial meniscus. You can see on the left, I have a diagram here that medial meniscotibial ligament is transected in the animals. 2 weeks later, they will receive an intra-articular dose of your test article, MOTYS, saline as a control, or a steroid-treated group as well. Some groups of animals also got a second dose at 4 weeks after surgery. And then we took it out to 8 weeks post-surgery before we did histology. So during the in-life portion of the study, we did weight-bearing pain measurements. So those injured joints, the animal will start to shift its weight to the opposing joint if it's painful, and then it comes back to normal if you relieve that pain. It's pretty standard incapacitance testing. It's commonly used. And then, as I mentioned at the end of the study, we can do histology and look for the different levels of cartilage degeneration and changes in the bone as well and score that. So a lot of data on this slide, but just to summarize it, I'll step through it fairly quickly. The top graph on the left is the single dose of MOTYS or saline or steroid. Bottom is repeat dose. The black line is osteoarthritic joint that's treated with saline. So this is the difference in weight bearing, the shift in weight to the opposing leg. You can see that that level of indicator of pain is increasing as the time goes on. Weeks after the dosing protocol here on the x-axis. And then both a high dose that we tested and a low dose of MOTYS can significantly reduce those pain levels, although it does come back as time progresses and the joint continues to be very painful and disease is progressing as well. And interestingly, the effects are pretty much equivalent, if not slightly better than a steroid treatment. And then repeat dosing kind of shows essentially the same type of data. On the right is just the area under the curve over the entire 6 weeks for those graphs. And you can see very significant reductions in the weight bearing difference for all the treatment groups relative to the saline treatment. And then finally, the histology, what that looks like. Top panels are, we've got a non-OA joint, so a normal rat joint shown here. This is stained with toluidine blue, so the very, very dark staining is the articular cartilage at the surface of the joints. If we treat such a joint with saline, you can see the development of a very severe lesion that's penetrating all the way down to the subchondral bone. And then treating with MOTYS, this is a high dose treatment, you can see pretty evident preservation of the articular cartilage at the surface. So not total prevention, obviously, but looks like we're at least having a significant delay or reduction in that progression of cartilage loss. And then just some of the parameters are quantified on the bottom here in the bar graphs. Cartilage degeneration score, which looks at the width of those lesions, how far they penetrate, what the level of staining is. Very significant reduction in that score. Total joint score also incorporates osteophytes. And so again, that's a very significant reduction in those animals. So just a very quick summary, some of the characteristics of MOTYS, it's amnion, chorion, and umbilical cord. It's micronized, lyophilized. There's no viable cells here. We've optimized a lot of the processing and characterization. It'll be available as a sterile product. It's readily resuspended in saline. We believe it has a multifactorial mechanism of action, which I think is going to be important in the treatment of osteoarthritis. Again, doesn't seem like there's a magic bullet. I've been involved in a lot of different programs throughout my time at different companies, and the magic bullet doesn't seem to work. We're going to have to target multiple pathways, I think, to make real progress in the treatment, certainly in the disease progression of osteoarthritis. These animal studies we've conducted, at least in a rat model, have shown significant reductions in pain and cartilage damage. And then some ongoing studies that we have going now, testing MOTYS in a larger animal model. It's work that we're now getting started with Dr. Lisa Fortier. She's at the Cornell College of Vet Medicine, and Larry has really helped facilitate that. Larry and I have collaborated a lot in the past, and hoping that as we kind of get things going here, we'll have some more stuff to collaborate on. Also looking at MOTYS versus some of the other biologics that are commonly used, and probably the most common in the clinic is platelet-rich plasma, or PRP. You'll see this even in the media these days. Golfers are getting treated with it, other professional athletes. This is a autologous treatment, so it's the patient's own blood. It's then taken out, and then it goes through a process that is extremely variable, centrifugation to concentrate the platelets, which are sort of vesicles containing natural growth factors, but a lot of variability there. So we're trying to do some of these comparative studies as well as we develop MOTYS. And then just one final slide I put on here, which I thought was quite interesting. Some of the work that we're doing looking at another very important aspect of osteoarthritis osteoarthritis is you could probably develop the best treatment in the world. You could inject it locally in the joint where it's going to have the most effect, but if it doesn't stay there long enough to have an effect, it's not going to do much good. So we did some work. Again, we collaborated with the Loser lab at UNC. We took the PTP or the MOTYS and fluorescently tagged it with a vivo tag, near infrared tag. This is an idea of what some of these particles look like that have been tagged with this tracer. And then we can use optical imaging like IVIS or AMI. Both of those units are available there at the imaging core facility, and do quantitative imaging in life of the animals over a period of time. So you can see right after injection, we've got a really nice, very localized hotspot within the rat joint, and we can follow those out over time. And some of the data we're seeing is quite an extended persistence of these tissue particles within the joint, and potentially that could occur in other tissues or organs of the body, even out to many weeks and potentially months after treatment. So I will stop there and gladly have discussion and questions. Thanks very much.
Thanks, Carl. That was great. Maybe I'll ask the first question as other people come on. So you mentioned the kind of regulatory environment and how that affects what you do as a discovery and a development scientist. So if there is a change in how these products are regulated, is that in some ways also an opportunity in terms of how the products can be developed? So what I mean by that is, if these are now going to be more regulated by drugs, or if the bar for minimal manipulation is more stringent, and you're already above that bar, does it make sense to think about additional manipulations that can control binding, that can control release rates, that can really let you tailor what is, a really sophisticated natural drug delivery system, right? If you were to try to design a drug delivery system that can deliver this many proteins in this particular pattern, it would be incredibly difficult. But nature's already done that for us. What are your thoughts on trying to engineer what you have now to get better binding profiles or better release profiles?
Yeah, I think it's a balance, Larry. Once you sort of commit to the FDA pathway, you more or less have to have defined what your product is going to be. Otherwise, you start to make changes, you're going to have to go back and do studies again, right? So it's a little bit restrictive in that regard, but I think you're right. If you can have this sort of foresight to say, is there something I could tweak here, at a relatively early stage that might make sense, then you should definitely do that. It's a whole area in itself, and a lot of these biologics conferences are having sessions devoted to this sort of somewhat confusing issue, right? This transition from a tissue product to a much more regulated environment. And a lot of the caution, quite frankly, has come from, and we've seen them ourselves, you've probably seen them on billboards as you're driving down the highway, stem cell clinics, right? That's and a lot of the safety issues that have occurred there. And it's more a case of, even though these particulate type of preparations have been on the market for quite a few years now, it's sort of recognizing that, we probably can't have all these multiple categories. Therefore, we've got to make one set of rules for these types of potential biologics. But I think your point of, again, back to that kind of balance of saying, here's what I think I should do, let's do it. And then but once you're committed down that regulatory pathway, there's so many, stringent criteria that you have to adhere to. If you try and make a change midstream, you're almost having to go back to the start.
Yeah, fair. Other questions for Carl?
Matt.
Okay.
Matt.
I have a quick question, Carl. What were the data? You called it a hotspot. What are those data at your last slide?
So that's taking the labeled tissue particles and injecting them directly into the rat joint, and then using, for example, the IVIS imaging system. So that imaging system is big enough to accommodate an entire rat, and then it's optical imaging. I'm sure the core facility, any university core facility certainly will have this optical imaging capacity now.
But the colors, it said, I'm just curious what the color scale is on that.
So that's just the intensity of the signal.
I see.
Fluorescent signals. And the darker red, I think it is, that's a more intense, that's a quantitative measurement of the amount of material that you're measuring.
I see. Okay, thank you.
All right. Thanks again, Carl. I think we'll move on to